Assalamu alaikum, dear students. I am Muhammad Rauf, lecturer, Department of Zoology Cast. Welcome to the course Principle in Animal Life 2. This is lecture number 17. The topic is X chromosome inactivation. Discussing agenda for the topic is X inactivation in female mammals. Lecture outcome. After watching and listening this lecture, students will know about X chromosome inactivation and its significance. X inactivation in female mammals. Female mammals, including human females, inherit two X chromosomes, twice the number inherited by males. The sex chromosome complement of the human male is XY, means the humans males are XY for their sex chromosomes, whereas the human females are XX. The human females carry two X chromosomes as compared to the one carried by the human males. So you may wonder if female makes twice as much of the proteins encoded by X-linked gene. Here you will start thinking about the transcriptional product of those genes. Means there is only one X chromosome in the human male. The Y chromosome only is responsible for determining the fertility and maleness, and X chromosome carries more than 1000 genes. So the human male carries a single X chromosome where only 1000 genes are present. A human female carries two X chromosomes where they have twice the number of genes for the X chromosome as compared to the males. So what will happen? Either it will have any harmful effect on the human females, or there is a way to compensate the dosage of X-linked genes and equalize the transcriptional product of X-linked genes in both cases. X-linked genes means genes located on X chromosome. In fact, almost all of one X chromosome in each cell in female mammals become inactivated during early embryonic development. Means a female is not even born yet. If that female is developing inside her mother's womb, and this process of X chromosome inactivation starts, where one of the X chromosomes in every cell is inactivated, irrespective of whether that X chromosome has been received from the father or from the mother. So in each and every cell, one of the X chromosome is inactivated. So we will have a mosaic-like situation. There will be some cells in the female's body where the X chromosome received from the father is inactivated. There will be other cells in her body where the X chromosome received from the mother is inactivated, so that females are a kind of mosaic for the X chromosome inactivation. As a result, the cells of female and males have the same effective dose. So when one X chromosome in the female is inactivated in each and every cell of her body, so it means there is only one functional X chromosome in females, just like one in males. The inactive X in each cell of a female condenses into a compact object called bar body. So one X chromosome is functional, where the other X chromosome becomes condensed and it becomes inactivated, and this inactivated darkly stained body is called bar body after the Canadian anatomist Murray Barr, who discovered them. So most of the genes of the X chromosome that forms the bar body are not expressed. So the inactivated X chromosomes genes are not expressed. In the ovaries, however, bar body chromosomes are reactivated in the cells that give rise to eggs, resulting in every female gamete having an active X after meiosis. So, at the time of gametogenesis, oogenesis, when the oocytes are formed, then both the chromosomes are activated. Why? Because the process will eventually form haploid germ cells. So, in the haploid germ cells, both the chromosomes needs to be activated. If the process of oogenesis starts in an ovary cell where one chromosome is inactivated, X chromosome, and the other X chromosome is activated, then the germ cells, some of the germ cells will have the inactivated X chromosome, others will have the activated X chromosome, so there will be a problem. So during oogenesis, even the inactivated X chromosome is reactivated. British geneticist Mary Lyon demonstrated that the selection of which X chromosome will form the bar body occurs randomly and independently in each embryonic cell present at the time of X inactivation. Just discussed earlier that which of the X chromosome will be inactivated, either the one received from the father or the one received from the mother. So that decision is random and independent, means in some of the embryonic cells the X chromosome received from father will be inactivated. In some cells the X chromosome received from the mother will be inactivated. As a consequence, female consists of a mosaic of two types of cells, those with the active X derived from the father and those with the active X derived from the mother, means some of the cells will have that chromosome inactivated which was received from the father, other cells will have that chromosome inactivated which was received from the mother. So there will be a mosaic type of pattern. After an X chromosome is inactivated, but remember one important thing here, that at the time of X chromosome inactivation, as we studied in the last slide, that this decision is random and independent, taken by every embryonic cell. But once a cell has inactivated a particular chromosome, suppose a chromosome from the father, then all the descendants of that particular cell will have the same chromosome inactivated which was received from the father, and same is for the mother. Thus, if a female is heterozygous for a sex-linked trait, about half of her cells will express one allele while the other will express the alternative allele. Suppose there is a sex-linked trait and that sex-linked trait is dominant and means if only one copy is defective, then it is expressed. But now the situation is that The female has one X chromosome inactivated. Suppose the female received a defective X chromosome from the father. Now that defective X chromosome will become inactivated in some cells and it will be active in other cells. So there will be some cells in the female's body where that particular trait will be expressed, and there will be some cells where that trait will not be expressed. In the next figure, we will see an example. Even in humans, there is an example in the recessive X-linked mutation that prevents the development of sweet glands. X-linked mutation. So means if the defective chromosome is received from the father and it is expressed in some cells, in other cells it is inactivated, so there will be cells in the female's body. In some cells the problem will occur, whereas other cells will not have such issue. A woman who is heterozygous for this trait has patches of normal skin and patches of skin lacking sweet glands. Very interesting situation where suppose the defective X chromosome is active in some genes, then those cells will lack the sweet glands, and if that X chromosome is inactive in some cells, then they will have normal skin. And usually what happens, the how the process of inactivation is done. Inactivation of an X chromosome involves modification of the DNA and proteins bound to its called histones. So the DNA is in association with histone proteins, and some modification occurs to those histones. Histones themselves are polypeptides. They are made up of amino acids. So, and they have a specific region called histone tails, where different types of chemical reactions called acetylation or methylation occurs. Methylation is usually a process where methyl groups are attached to the histone tails, and it strengthens the association between the histones and DNA, and as a result the chromosome becomes inactive. The condensation is increased, and this particular chromosome or area of genome becomes inaccessible to the transcriptional machinery or DNA polymerases. So several genes are involved in the process of inactivation. This process is still a mystery and a lot more needs to be explored about it. Here you can see an example. Now this is early embryo. That cat received that particular chromosome from father and that particular chromosome from mother. Now in the early embryonic stages, one of those X chromosomes will be inactivated, and it will become stay inactivated in all the descendants of that cell, and the decision to inactivate a particular chromosome will be random and independent. Some of the cells, this particular chromosome will be inactivated. In other cells, this particular chromosome will be inactivated. So Then you can see here a mature cat where there are cells in which the chromosome for orange fur is inactivated, and there are cells in which the chromosomes for black fur is inactivated. So you can see here the in the cells which are present in that area, the allele for orange fur is inactivated, and here you can see black color. Here in that particular area, a cell exists where the X chromosome having allele for black fur color is inactivated, and the fur color is orange. So it provides a perfect example of the mosaicism of X chromosome inactivation. So one of the genes called ZIST for X inactivation center becomes inactive only on the chromosome. Now here what we know about X chromosome inactivation, that there exists a particular gene called ZIST on the X chromosomes, and the X chromosome that is destined to be inactivated, that ZIST gene is activated on that chromosome which is destined to become inactivated. Multiple copies of the RNA product of this gene apparently attach to the X chromosome, on which they are made eventually almost covering it. So that ZIST produces several copies of an RNA, and then this RNA covers that particular chromosome on which that active zest gene is present. Interaction of this RNA with chromosome initiate X inactivation, and the RNA products of nearby genes help to regulate the process. Thank you very much. Allah Hafiz.